Hello class, our today's topic is cell, the unit of life. In this chapter, we are going to study about the eukaryotic cell, prokaryotic cell and the cellular organelles that present inside the cytoplasm. So first let's discuss about the what is cell. Cell is known as the structural and functional unit of the living organisms. Means entire body of any living organism is made up of cell, and cell is going to perform all the vital function of any living organisms. So in the system we find that cell is the structural and functional unit. On the basis of the number of cell present in any living organism, means if any living organism which is made up of only one cell, means the entire body is of only one cell, it is called the unicellular organism, like amoeba, paramecium, plasmodium, etc. And if the organism is made up of more than one cell in their body, it is called the multicellular organism. Like a human being also, fish, frog and all the higher category and group of the animals and plants. This term cell is first coined by the observation of the Robert Hooke, who described the living cell in a cork cell of the plant tissue. But the cell theory means what is the definition is going to be studied as a theory of the cell, means all the living organisms that is composed of the cell and the cell products, and all the cells arises from the pre-existing cell. Actually this term that all the cell arises from the pre-existing cells is given by the Rudolf Virchow in 1855. He has explained that going to say that omnis cellula e cellula, means each cell is originated from the pre-existing cell. There is no pre-existing cell, so O means the new cell is formed always from the presence of the pre-existing cell. And this cell theory is given by the Sliden and Swan in 1838-39, and by this discoveries the scientists performed the cell theory. So if we are going to study about the cell, there is a two type of cell, means we know that plant cell and the animal cell. In the plant cell, the overview is that the outermost layer of plant cell is called the cell wall, which is made up of the cellulose, and it is found on the outer surface of the plasma membrane. One more important characteristics is found in the plant cell, that in the plant cell there is a presence of plastids. Plastids are those organelles which contains the pigments and are very characteristics, like if the green color, it is known as the chloroplast, if colorless, that is the leucoplast, and if it is red or orange color, it is chromoplast. Centrioles are actually absent. Hence the mitosis is anastral, means no centriole is there. Mitosis division is found with the help of a plate formation in between the two cells, and this is known as the mitosis plate, and vacuoles are found in a very large number and having a very large size also. The Golgi bodies occurs in a distinct manners unit, and it is known as the dictosomes in case of the plant cell. This is the 3D vision or the 3D structure of a typical plant cell. You can see here the outer areas is having a very thick cell wall that is made up of the cellulose, and For the distribution, for the conduction of the material from one cell to the another cell, there is an opening is present in between the two cell wall, that is known as the plasmodesmata. And all the living organelles like Golgi bodies, Golgi apparatus, the mitochondria is there, nucleus is also there, the vacuoles is also there, and then ribosomes, lysosomes, all are present in the cytoplasm. So this is the typical structure of a plant cell. If we are going to study the overview of animal cell, there is a no cell wall is present in the outermost layer, and no plastids is also present in the animal cell. But the centrioles are present, and it is a distinct, and mitosis is ancestral, means the cleavage is formed for the division, and the vacuoles are either absent, or if present it is very small and in a very few number. The Golgi bodies are elaborated forms, but it is interconnected with each other. In this diagrams, you can see the different shapes of the animal cell. You can see here the oval shape, the cell of arterial wall, and then the melanocytes cells are present in the skin. Human X cell is also there. Human RBC, which is also non-nucleated. Nerve cells are very long cells, striated and unstriated muscles fiber, and there is a sperm cells also, ovum cells or egg cells is also, skin cell is also. So you can see here all the cells having different shape and size according to the position of where it is found. This is the typical structure of a animal cell. You can see here no cell wall is there, only plasma membrane is there, but the Golgi apparatus is also there, cytoskeleton is also there, lysosomes or the suicidal bags is also there, but one very unique characteristic that the centrioles or the is present in the central portions, which was absent in the plant cell. On the basis of the nucleoids, or you can see the DNA present in the cytoplasm, whether it is in a membrane bond structure or not. The entire cell is differentiated into the prokaryotic cell and the eukaryotic cells. The prokaryotic cells are generally smaller in size and multiply very faster than the eukaryotic cells. But on the basis of their shape, the prokaryotic cells is of 4 types. You can see it is a rod. If it is rod like means bacillus bacteria, if it is a spherical coccus bacteria, comma shape it is a vibrio cholerae cholera bacteria, and sometimes it is found in the spiral shape, is known as the spiralum. So these are the 4 basic structures or the shape of the prokaryotic cells. The nucleus having totally lacking in the membrane, and it is not well defined, that the genetic material is basically naked. So in addition to the chromosomals or the genomic DNA, many bacteria contain a smaller circular DNA in the cytoplasm. Mostly the bacteria have a circular DNA in the cytoplasm, and this is known as the plasmid. A plasmid having a genes for the certain very unique characteristic like resistant to the antibiotics, or sometimes the sex factor is also. So these circular DNA or the plasmids have resistant genes to the antibiotics. The cell membranes that form enfolding cell membrane is called the mesosomes. You can see here in this picture the cell wall is having a three wall structures. Pili is also there for the joining of the 2 bacterial cells. Flagella is also present for the locomotion, and the extracircular green color you can see here, the genetic material is known as the plasmid. This is the structure of a prokaryotic cell. Plasma membrane is there. DNA is also present at the center. The nucleoid region is also there, and for the locomotion purpose the flagella is present, and for the joining of the 2 cells for the purpose of the conjugation, and or you can see in the sexual mode of reproduction, the pilis is also present. This is known as the pilis or pilas is there. Cell envelope of a prokaryotic cell having a tightly bond three-layered structure. The outermost is made up of the glycocalyx, made up of the glycolipids, and the middle is the cell wall, and inner is known as the cell membrane. The glycocalyx, or you can say the outermost layer of a cell, serves the two basic functions, means it is going to be provide the protection as well as going to the adhesion of the two different cells. And the cell wall is made up of the peptidoglycans, that made up of the repeating unit of the N-acetyl glucosamine, that cross-linked by the short peptide chain. Based on response of the cell wall to the staining techniques, means if we are going to stain the bacterial cell, and if it is, and after the washing with the alcohol, it is going to be retained or sometimes washed away. The bacterial cell is of two types. One is the gram positive, another is the gram negative. The gram positive bacteria are those bacteria that retain a purple color or the blue color after washing with the alcohol. Reason behind this, that the cell wall is very thick, about 20 to 80 nanometer in the thickness, and it is made up of the peptidoglycans, which is the composition is 70 to 80% of the cell wall. Mesosomes are in abundant form, and it is going to be resistant to the lysozyme action. In gram negative bacteria, it do not retain any color after washing with the alcohol, so do not retain color. Reason behind this, the cell wall is a very thin, having 8 to 12 nanometer in the thickness, and the peptidoglycan is about the 20 to 30% only present in the cell wall. Mesosomes are less abundant, and it is very susceptible to the lysozyme, means the enzymatic action. The cell membrane or the plasma membrane of the bacterial cell having that boundary for the cytoplasm, and it is a semi-permeable. Semi-permeable means it allows few chemical compositions to travel from one cell to another cell. Then it is made up of the lipids 20 to 30%, protein 50 to 70%, and oligosaccharides having 1 to 2% only, and 20% of the water. These lipids are mainly in form of the phospholipids, means phosphorus and lipids. Glycolipids mean glycogen, glucose or lipids, and sometimes in a very complex structure it is known as the cholesterol. The membrane lipids are amphipathic, that they have both the amphipathic means they are both the nature hydrophilic and hydrophobic. Hydrophilic means water loving, and hydrophobic water hating. The protein exists as the transmembrane protein, means example, it is made up of glycoporins and extrinsic proteins, that is known as the spectrin, and intrinsic proteins is also there. The plasma membrane extend into the cell wall in form of the vessels there, sometimes in the very tubule-like structure, that is known as the lamellae. Lamellae is called the mesosomes, and mostly very common in the gram negative bacteria, and due to this region, the gram negative bacteria are more pathogenic in comparison to the gram positive bacteria. There is the chromatophores and some nucleides and flagella is also there. The internal membrane systems containing the pigments, and these are the extensive in the photosynthetic bacteria and cyanobacteria. Cyanobacteria means blue green algae, and photosynthetic bacteria, that bacteria prepare their food. Nucleides is a singular and circular DNA which does not having any histone protein, no histone protein for the binding of these segments. And the flagella, if present in the bacterial cell, is provided with the filamentous extension, and it is the purpose of is for the motivations, or we can say it in a motile forms or movement. The pili and fimbris is also present in the bacterial cells to attach the host tissues, and or to be any solid surface like rocks in the streams. The ribosomes and elutions body is present in this cell. The ribosomes are found attached to the plasma membrane of the cell, and the ribosomes units are 50s and 30s, means 70 types of the ribosomes unit, and some inclusions bodies like a non-membranous bond structure that freely found or lying in the cytoplasm, which serves as a reserve food material like phosphate granules, glycogen granules, and sometimes cyanophyceans granules also. These are known as uh, the structure made for the reserve food materials. So now the eukaryotic cells. In the overview of the eukaryotic cells, we find that those cells which have a nuclear membrane and other membrane bound organelles, all the organelles having the membrane bond. The cell wall structures is composed of the lipid bilayer, means two layer of the lipid and one layer of the protein in the cell wall. And due to this structure, it is known as the fluid mosaic model, which is given or proposed by the Singular and the Nicholsons in 1972. In the membrane transport formations, the most important function of the cell membrane is transport of the molecules across the one cellular membrane to the another cellular membrane. And there are two methods of the transport. One is the active and another is the passive transport. In active transport, there is a utilization of metabolic energy. Means with the utilization of energy, the movement is there, the transportation is there. It can occur even against the concentrated gradient sometimes, and it is a very rapidly process. This process is affected by the absence of the oxygen sometimes, cyanide, and if there is a very cold temperature. In case of the passive transportation, there is no expenditure of any kind of energy, and it occurs only in the following concentration gradient, because it is a very slow occurring process and it is not affected by unavailability of the oxygen or cyanide or the temperature. So this is all about the cell structure and functional unit. In the next portion, we will study about the cellular organelles. Thank you and have a nice day.